Hello everyone. In this session we have to deal part 2 of chapter 8, that is cell the unit of life. The content to be discussed in this part, that will be structure of a eukaryotic cell, how does a plant cell differs from animal cell, then we have to see detailed structure of a plasma membrane, then most accepted model of plasma membrane, functions of plasma membrane, and then some characteristics of cell wall found in eukaryotic cell. This is part 2, chapter 8, cell the unit of life. In this we have to discuss detail of eukaryotic cell. I told you that is in biological classification. The eukaryotes include the protist, plant, animal, and that is fungi. Prokaryotes, they were included in kingdom Monera. Rest of the four kingdoms, they have eukaryotic cells. What mean by extensive, that is compartmentization is there in cytoplasm through presence of membrane bounded organelles. That this is the cell here, this is one compartment nucleus, that this is mitochondria, that this is endoplasmic reticulum, that this one is Golgi body, that this one is plastid. So in a cell, that is differently compartments are there. Eukaryotic cell, as you know, it is having an organized nucleus, and this is having a nuclear envelope. It is separated from the cytoplasm. It is connected only through certain pores. Eukaryotic cell, that is having variety of complex locomotory, and that is cytoskeletal structure. Detail of that we have to see in time to come. Their genetic material is organized in two chromosomes. Chromosomes are there. In case of prokaryotic cell, chromosomes, that is chromatin material, was not organized. Chromosomes are found only in case of eukaryotic cell. All eukaryotic cell, these are not identical. The basic plan is same, but they are not exactly identical one. The eukaryotic cell we can divide, that is plant cell and animal cells. We have to see what are the differences. Plant cell and animal cells. Plant cell, it has a cell wall, it has a plastid because it has to do photosynthesis, and it has a large central vacuole, three important characters which are absent in case of animal cell. On other side, we see that such things are absent in animal cell, but in animal cell, one organelle we have, that is centriole is present in animal cell, which is absent in case of that is plant cell. You can see that is beautiful colorful diagram we here, plant cell and animal cell. You can see in plant cell start from here, that is from where we have to start. You can see here, this is the cell wall. Cell wall is found only here in this case. That is here we have, this is the chloroplast. This is found only in case of plant cell. That is a large central vacuole is there, and the middle lamella is present only in case of plant cell. Rest of the structure you are seeing here. Detail of this we have to see in time to come. But you see in case of animal cell, that is, it doesn't have cell wall, it doesn't have middle lamella, it doesn't have a large central vacuole. The vacuoles, if are present, then that will be of small size. And very important one, that is the centriole is there, that is near to the nucleus. If I am going to make, this is animal cell, that this is that is nucleus, that is close to the nucleus. There is centriole, where two centrioles are there, which are perpendicular arranged, and that make the centrosome. And this centriole, that is absent in case of plant cell. Is it? Rest of the structure, we have to go in detail of this, then you can compare it. Okay. Now the same thing we have here. In case of plant cell, usually these are larger, these are smaller. That is cell wall is present in addition to plasma membrane, and it consists of middle lamella, primary, secondary cell wall. Here the cell wall is absent. Plasmodesmata, these are present. Plasmodesmata, what we mean by that, this is one cell, that this is the other cell here. This is that is middle lamella, that is the cytoplasmic connections are there in between that adjacent cell known as plasmodesmata. Here plasmodesmata are absent. That is chloroplast is there, plastids are there, vacuole large and that is permanent. Here the chloroplast is absent, vacuole small, and these are temporary one. Around the vacuole, a membrane is there known as tonoplast, because the vacuole is absent here, tonoplast is absent. Very important point, centriole is absent except in motile, that is lower plant cell, that is the zoospore or the gametes are there. Otherwise, that is centriole is absent in case of that is plant cell. it is present here. Nucleus present along the periphery, because of that is central vacuole, that is this is central vacuole. The cytoplasm and the nucleus is shifted towards periphery while the nucleus is almost at the center. Lysosomes, you have to see the function. These are rare. These are very common in case of animal cell. Storage material is in the form of starch, because starch will be formed during photosynthesis, while in case of animal cell, the storage material is in the form of glycogen granules. Is it right? Come to the next now. We have to see now cell membrane, plasma membrane. We have to see plasma membrane. Either this is in prokaryotic cell or this is in that is eukaryotic cell. The structure is almost same. Detailed structure of the plasma membrane was studied only that is after that is advent of electron microscope in 1950. You see, chemical studies of cell membrane, especially we have studied that is human are basic. It that is enabled the scientists to deduce that the possible structure of the plasma membrane, that is chemical studies, they have revealed the structural of plasma membrane. These studies they show that the plasma membrane is mainly composed of lipid and protein. The major lipid that is phospholipid. These are arranged by layer. You see here that these are the molecules of lipids. That is, this is bilayer is there, lipid molecules, and that is their polar head. This is polar head, that is towards outer side or inner side, and that is hydrophobic tail is towards, this is the hydrophilic tail, that is inner part, that make the inner part. This ensures that the non-polar tail of saturated hydrocarbon is protected from aqueous environment. In addition to phospholipid, that is the membrane may also have that is cholesterol, and then this is phospholipid. These are the important phospholipid compound. The biochemical investigation that is clearly revealed that the cell membrane also possesses that is protein and carbohydrate. The ratio of the protein and lipids, it varies considerably in different type of cell. You see, this is the bimolecular lipid layer, and the proteins are found that is on outer side, inner side. Some of the protein molecules are found here in between the lipid molecules also. See, that is various models have been proposed. That is how the protein and the lipids these are being arranged. We have to see now. See, chemical composition. What are the chemicals usually found in that is plasma membrane? The protein is there. That is proportion of protein and lipid that will vary. Lipids are 20 to 79%. Carbohydrates are comparatively very low, 1 to 5%. About 30 enzymes are found, and water is about 20% of the total weight. Important question. In human being, the membrane of RBC erythrocyte that you have seen approximately 52% protein, and that is 40% of lipid is there. You have to remember this question has come that is many times in examination. Depending on the ease of extraction, membrane protein, you see that how we can extract, how can we separate the protein from lipid in plasma membrane. Depending upon the ease of extraction, the membrane protein are classified into two, that is integral and the peripheral one. You see that this is the plasma membrane. I can make that these are the lipid molecules like this. This is the polar head, this is the non-polar head. These are the proteins which are found at the surface, either inner side or that is outer side. These can be separated easily. These are known as peripheral protein. These are called peripheral protein, can be separated easily. While the integrated protein, these are embedded inside the lipid layer, and that will be rather difficult to separate these. These are known as integral protein, right? That is, integral protein are partially or totally buried in the membrane in between the lipid molecules. Is it right? This is a beautiful diagram of NCERT we have here. You see how this protein and lipid molecules are being arranged. Various models have been proposed. That is, out of these so many models, the most that is accepted model is that is fluid Moisic model given by that is Singer and that is Nicholson. What we have in this model, you have to see here, you have to see that I have to explain it first. That is, as I told you earlier also, that is, these are the lipid molecules. This is the surface. On surface we have carbohydrate in the form of sugar. This is the surface on. This is the peripheral protein we have here. That is, this is the cholesterol we have here. This is the protein which is remain embedded inside the, that is lipid layer, integral protein, and these are the phospholipid bimolecular layer. Is it right? That is simply you can understand that these are the, that is lipid molecules, bimolecular layer, again that is repetition of this, and these are the, that is peripheral protein, either this side or this side, and these are the, that is integral protein. That is, here we have, that is sugars or carbohydrate attached to this. Sometimes there may be, that is cholesterol found in between the bimolecular lipid layer of phospholipids. Is it right? Now, the most, that is accepted model of the plasma membrane is, as I told you, 𝕆𝕋𝔼𝔼 model. This is improved model structure of the plasma membrane was proposed by Singer and Nicholson, that in 1972. It is widely accepted one. According to this, quasi-fluid nature of lipid enables lateral movement of protein within the overall, that is bilayer. That is, here the lipid will make the C, that is lipid molecule, they will make the C, and the protein molecules, these are just like, that is the iceberg. This iceberg, sometimes these are here, sometimes they will go more deep, sometimes they will come on the surface icebergs. That is, this ability to move within this membrane is measured, that is fluidity of membrane. Fluid nature of the membrane is also very important. Why it is important? Because that this can correlate the function of the plasma membrane, that is the cell growth, the formation of intercellular junctions, secretion, endocytosis, and cell division. These are the important function of the plasma membrane. They can be explained only on the basis of when we follow that the model of, or the plasma membrane is, that is having this fluid model. Functions of the plasma membrane you see now. One of the most important functions of the plasma membrane is the transport of molecule. This is the cell, that this is plasma membrane. What will go inside, what will go outside, that is being controlled by plasma membrane. So this is very much selective in nature. Transport of the molecules inside and outside will be controlled by plasma membrane. This is the function. Now we have to see how these will be, that is transported. How, that is, the things will go inside or outside. You have to see now. That is, here we have to see passive transport and active transport, two important features of the plasma membrane we have here. What in by passive transport? You see that this is the plasma membrane we have here. This is plasma membrane. This is selective permeable to some of the molecules present on either side. That this is the, that is outer side, that this is the inside. Many molecules they can move, that is in membrane without requirement of energy. This is called passive transport. Neutral solute may move across the membrane by the process of simple diffusion along the concentration gradient. What we mean by, you have to study that detail in chapter transport in plants. You see here that the things which are soluble in water will be rather difficult to cross the membrane. The substances which are soluble in fat, because it is made up of lipid, that's why the fat soluble substance, they can move easily, that is through plasma membrane. This one is plasma membrane. See what happens, if the concentration of these substances is more this side, then from high concentration to low concentration, the fat soluble substance they can move easily. It is, that is according to along concentration gradient. But you see, substances which are soluble in water, the plasma membrane doesn't allow. Even if these water soluble substances, if are more this side, they cannot go, that is towards the low concentration side, because these are water soluble one. So here in plasma membrane we have some, that is carrier proteins we have here. This the carrier proteins, and these carrier proteins, that is they will facilitate that is diffusion of water-soluble substances from higher concentration to that is lower concentration. So this is known as what? Passive transport. This is called, come on, passive transport. Is it clear? This is known as simple diffusion. This is called simple diffusion when the proteins are not required, and when that is carrier proteins are required for water soluble substances, that is known as what? Facilitated that is diffusion, right? In both these are the types of that is passive, that is transport. Energy is not required. Why? Because it is according to concentration gradient. You can read it out. That is from higher concentration to lower concentration, substance will move. Water may also move across this membrane from higher to lower concentration. Movement of the water, you know, is known as that is osmosis. As the polar molecules cannot move through the non-polar lipid layer, they require carrier protein. Is it clear to you? That is known as facilitate, that is transport, facilitate passive transport. Am I clear? Should I repeat? Passive transport, 1 point, that is this is according to concentration gradient. From high concentration to low concentration, substance will move. Fat soluble substances, they will be easily facilitated through move from that is through plasma membrane, but the non-polar substances which are soluble in water, they will be allowed to move from higher concentration to low concentration only through carrier protein. Is it clear? Come on. Active transport, that is what happens now. Active transport, that this is the plasma membrane again. This plasma membrane, that here the concentration is low, here the concentration is high. When the substance they move from low concentration to that is high concentration, this is against concentration gradient. A few molecules, ions or molecules are transported across the plasma membrane either from this side to this side. That will always be from that is low concentration to high concentration against that is concentration gradient from low concentration to high. Are you getting my point? Such transport that requires energy, that is known as, that is it will go from that is low energy to high energy, and it is known as that is uphill movement. What we can call it? We will call it as that is uphill movement, and in this that is ATP will be consumed. That's why it is known as that is active transport. Such transport is an energy depending process, that is it will use ATP here, utilize ATP. It is called active transport. For example, we have sodium and potassium pump we have, that is which utilize energy. Detail of this you have to see in chapter that is transport in physiology, transport in plants. Is it right? Now we have to go for the next one, that is a cell wall. In case of plant cells, it is a non-living rigid structure. Non-living this is hard structure, rigid structure, which is the outermost covering, that is which is found outside the plasma membrane in fungi and in plants. Even in protist we have that is the cell wall is present. Algae has cell wall. It is having cell wall made up of cellulose, that is galactans and mannans, and the minerals like calcium carbonate, while in other plant it is made up of cellulose, hemicellulose, pectin, and the that is protein like substances. You have to remember it. Cell wall of the young plant cell, you see that this is the cell wall like this. This is the cell wall. That is beneath the cell wall we will have, this is cell wall. Is it right? That is here we have, this is the plasma membrane. So this cell wall is present since beginning, and this cell wall is known as that is primary cell wall. So primary cell wall is found just outside the plasma membrane, and this primary cell wall is found in that is all plant cell, that is young cell. Is it? This is what happens when the cell grows, then that is outside this plasma membrane. Now you see this is plasma membrane. The secondary cell wall will also be deposited. This is secondary cell wall, and that will be that is primary cell wall. This is primary, right below the primary cell wall, that will be the secondary cell wall will be deposited. Come on now. Cell wall in young plant cell, the primary cell wall is capable of growth, which gradually that it diminishes as cell mature, and the secondary cell wall is formed on inner side of the, that is inner side towards the membrane side of the cell. The middle lamella is found. Middle lamella is a layer in between the two cells. That this is one cell here we have, that this is the other cell we have here, that in between the two cells cementing material is found, which is known as that is middle lamella. This middle lamella is made up of calcium pectate, and it will hold the cells together. The neighboring cells will be kept together. The cell wall and the middle lamella may be traversed by plasmodesmata. I told you, this is cytoplasm of cell A, this is cytoplasm of cell B. How the cells are connected, that is in primary cell wall and in secondary cell wall, that is certain pores are there, pits are there, and through these pits the cytoplasmic threads are there, that is through which the neighboring cells are connected. These cytoplasmic connections are known as plasmodesmata. I told you pits also, that is this is the pit, this is the, that is pit. Through this pit the cells, neighbor cells, these are being connected. Pits are unthickened area. In this area the secondary cell wall is absent. In pit, that is secondary cell wall is absent, and only the primary cell wall is found. And here pits are usually found in pairs, some microscopic pores these are, and the cells are connected through this. Okay, you can see here that this is one cell, this is another cell, that here the area is there where we do not have, that is secondary cell wall is absent here, and this pore will be known as pit. Through this pit cytoplasmic connections are there. Is it right? Okay, now we have to see detail of this plasmodesmata. I told you, that is plasmodesmata, that you can see once more here, that you see that this is, I am going to make, this is one cell, that this is the other cell. In between the two cell we have, this is the, what is this? This is the middle lamella. What is this? This is the, that is primary cell wall. This is the primary cell wall we have here, that this is the secondary cell wall. You see the area where we do not have deposition of this secondary cell wall, this is this area where we do not have deposition of, that is secondary cell wall. Is it? What is this? That is primary cell wall. This one is, that is secondary cell wall. So this area is called, that is pit, where the secondary cell wall is absent, and this pit is usually in front of the pit of, that is neighboring cell, and through these pits these cytoplasmic connections are there. These cytoplasmic connections are known as, that is plasmodesmata. Come to this cytoplasmic, that is between adjacent plant cell, that is these are cytoplasmic threads which develops, that is in minute pores in the wall. It forms living component in the dead cell. A number of the plasmodesmata of the cytoplasmic stent are present in pit. These are the cytoplasmic stent, right, through which the cytoplasm of one cell is connected to the other. These are lined by plasma membrane which contain a fine tubular structure. This cytoplasmic thread, these are lined by, that is a fine plasma membrane, and then this thread-like structure, these are known as desmotubules. A tube-like structure is formed. This is plasma membrane, and this one is, that is cytoplasmic thread. Is it right? Endoplasmic reticulum, that play important role you have to study in origin of plasmodesmata. There the cell wall, you know, cell wall is non-living one, that is apoplast, while this plasmodesmata, because these are the living one, they make the, that is symplast. Symplast means that is living in between the two cells. Is it right? Come to the last part of this chapter, this part, function of cell. You see, in case of plant cell, because this is the hard and rigid structure, it will provide a definite shape, it will protect and give mechanical support to the plant. You know, animal cells, if these are placed in hypertonic solution, that is squeezing will be there. The plant cell, because of presence of rigid and tough, that is cell wall, that is it used to get a definite shape. It functions apoplast. Apoplast means that is, this is non-living part of the cell, is it? It forms skeletal framework of plant and provides mechanical support, the same thing, and its deposition like cutin and suberin reduce transpiration. What we mean by that is cell wall is there, that is leaf, epidermis is there, it is having the cell wall is there, and because this cell wall is having cutin and suberin, and because of this the water will not be transpired, that is, it will reduce the transpiration. The cell wall, because having cutin and suberin like substances which do not permit, that is, entry of water, or that is, the transpiration will be reduced. This is part 2, chapter 8, cell, the basic unit of life. Thank you.